In this video, we'll talk about the immune system, homeland security for our bodies. You should be able to state at the end of this video what the major components of the adaptive immune defense are against microbial and viral pathogens. And you should also have an understanding of how the adaptive immune system recognizes or is able to recognize so many different antigens it has never before seen. And based on this understanding of the how the immune system works, or how the adaptive immune system works, you should then be able to further explore such questions as how vaccination works and what the consequences of HIV infection are. So our bodies have a layered defense. All living organisms, essentially, have what we call an innate immune system. And now the innate immune system has a nonspecific response. It generally reacts against a broad range of viruses and bacteria. There is no modulation of the innate response. The innate response consists of things like physical barriers, acidity, antimicrobial peptides, phagocytic cells. And when they see a bacterium or a virus, they respond to it immediately and maximally. And there's no further fine-tuning or modulation of the response down the road. So the innate immune system does consist of both cells, such as the phagocytic cells, and humoral, in this sense, means not funny, haha, but consists of soluble factors. Okay? These could be peptides and antimicrobial proteins that are soluble and circulating in the system. And the innate system has no memory. Again, it has a very broad spectrum activity, and there is no need for an innate immune system to remember in particular that it has seen a particular strain of virus or bacterium before. And as I said, it's found in all cellular life. But that's really all we're going to say about the immune system. What this video is really about is the adaptive immune system. This wonderful defense system that mammals and other vertebrates have. Okay? The special characteristics of the adaptive system are that it is specific. Okay? So it is directed against a particular virus strain or a bacterial strain. Now, even though it is wonderfully specific, there is a cost to the adaptive immune system, or a disadvantage to the adaptive immune system, is that it takes some time for the body to launch an adaptive immune response, typically 1 to 2 weeks. So there is a lag time between when a virus or bacterium first enters the body and if mounting of an effective adaptive immune response against that particular strain. And it consists of both cells, active cells, and we will see these are T cells and B cells. And the humoral components, again, the soluble factors, and these are proteins, sometimes called immunoglobulins, but you probably are more familiar with the term antibodies. These are soluble proteins circulating in the body. The wonderful thing about the adaptive immune system is not only is it specific, but it remembers. It can remember that it has seen a particular virus or bacterium before, and the second exposure leads to a much stronger and much quicker response. So the memory is manifested as faster response, meaning the lag time is greatly reduced and it's much stronger. And it's immunological memory that allows us to vaccinate. So vaccines work on the principle of immunological memory, that the body, having seen a vaccine, will be able to defend itself very quickly and very strongly against any other organism that resembles that vaccine. So this wonderful adaptive immune system is highly evolutionarily restricted. It's found only in the jawed vertebrates. And this figure shows you essentially the broad spectrum of life. Here we have the single-celled eukaryotes, the protists, plants, fungi, animals. And here on the one small branch of the animal tree, the jawed vertebrates, outlined in red, these are the only organisms that have the adaptive immune system. Neither bacteria nor archaea have anything like that, but all of these also contain innate immunity. So here graphically is an illustration of the properties of the adaptive immune response. Let's say that at time 0 we have a virus that enters the body. So we have a virus infection. It takes a week or 2 weeks before we see antibodies against the virus circulating in the bloodstream. So this is virus strain A. And after and what this is tracking is antibody concentration. But if we were also looking at T cells, the T-cell response against the virus would follow a similar kinetics, would show a similar delay and similar kinetics. Once the virus has been removed by the body by both antibodies and T cells, the immune response goes down. The amount of antibodies and the number of circulating T cells goes down. Now let's say at this point, at this time, well, maybe about 29 days or so, we introduce a mixture of virus A and virus B. What we see is that with almost no perceptible lag or delay this time, there is a very strong response to virus A. Right? This is an antibody directed against virus A. So this is the manifestation of immune memory. The body remembers that it has seen virus A before and is able to mount a very rapid, very strong immune response. However, the body has never seen virus B before, so it still takes another week or so before the body can mount an immune response against virus B. And the antibodies to A will not react with virus B, nor will the antibodies to virus B react with virus A. So this illustration then shows both the specificity and memory of the adaptive immune system. What we call the humoral and cell-mediated branches are actually the antibodies. And I would like you to remember that antibodies are produced by B cells. There are lots of what we might call naive B cells that are sort of residing in the body, not doing much of anything. But once B cells are activated B cells turn into plasma cells. And plasma cells then are the product of B cell activation, and plasma cells are antibody factories. They will no longer divide, but they churn out great quantities of antibodies. And there are 5 different classes of antibodies. The most common ones are immunoglobulin G, that's found in blood and lymph circulating in the blood and lymph. There is an immunoglobulin A, found primarily in mucus and tears and milk and saliva. Okay, so this is sort of in all the secretory fluids that contain IgA. And then there are a few rare antibody types, and they have special roles, but we won't talk about that in this course. So these antibodies bind to bacteria or viruses, and this binding of antibody helps inactivate these viruses and bacteria, and also targets them for phagocytosis by phagocytic cells, which are actually part of the innate immune system. So there is coordination and cooperation between innate and adaptive immune system components. So that's the humoral response, or the humoral branch of the response. The cell-mediated branch depends on killer T cells. Well, nowadays all the textbooks call them cytotoxic T cells, but I really liked it much better when they call them killer T cells. And that's what they do. Their job is to attack and eliminate the body's own cells. What? Well, it's the body's own cells that have been infected by virus, have turned cancerous, or, and this is not something that would, a situation that would occur naturally, if there is a tissue graft, okay, and so you have foreign cells, then the cytotoxic T cells will kill and attack those as well. I think the presumption is that these foreign cells look suspiciously like cancer cells, and that's why the body attacks them. So let's explore how the adaptive immune system gets its specificity. The specificity arises from proteins that recognize what we call antigens. And so there's two special words here. Well, antigen here is the special word. Okay. So antigen is any substance that can bind to antigen receptors. It's kind of like a circular definition, isn't it? But antigens also generally are able to provoke immune responses. But you know, as circular as this definition is, it's essentially any substance that can bind to the body's antigen receptors. So what are these antigen receptors? This is an antibody molecule, or immunoglobulin. This is sort of the basic immunoglobulin type subunit. It consists of 4 protein chains. 2 heavy chains, so these are heavy chains that I've labeled H, and these are identical to each other. So these are homodimers. The 2 heavy chains are identical to each other. And then there's 2 light chains. And the light chains are also identical to each other. So in fact, if we split this antibody molecule in half, the left half is identical to the right half. They're mirror images of each other. These lines here indicate disulfide bonds that link these polypeptide chains together into this coordinary structure using covalent disulfide bonds. Now if we look at these colors, you see that these parts of both the light chains and the heavy chains have a different color. They're colored blue. And these are what we call the variable domains. And that's because the parts of the molecule colored brown or maroon, depending on your monitor, these are constant domains. For a given class of antibody, all the antibody molecules have the same constant domain chains. But if you look at different antibodies that bind to different antigens, what we see is that they vary in these variable domains. And in fact, the variable domains are at the N-terminus, the constant domains are at the C-terminus. And this is true for both the heavy and light chains. Okay. The N-terminus is the N-terminal part of the polypeptide chain consists of the variable domain. So the variable domain is called variable domain because there is actually variation in the amino acid sequence in the variable domain. And there's one part of the variable domain, these tips, which are hypervariable. They're even more variable than the rest of the variable domain. And they have the really fancy name of the complementarity determining region, or CDRs. Suffice it to say that this is the business end of the antibody molecule. It's this hypervariable or CDR regions that actually bind to the antigen. So each antibody molecule has a particular antigen binding surface that is comprised, that is made up of a light chain and a heavy chain variable domain, and these two together form a single antigen binding pocket or surface. Okay? And the variability of the amino acids in this region explains their variability in terms of antigen specificity. And these are found on the surfaces of B cells. Okay, so immunoglobulins are also actually the B cell antigen receptors. So the B cells have on their cell surfaces, on their plasma membranes, a membrane-bound version of immunoglobulins that serve as their antigen receptor. T cells, the other major cell class of lymphocyte or immune system cell in the body, also have their own antigen receptor, and this is called the T-cell receptor, or TCR. And the T-cell receptor consists essentially of these two chains. There's an alpha chain and a beta chain, and the two chains together make up a single T-cell receptor. Again, the chains that make up the T-cell receptor consist of a variable domain, and here they're not differently colored, unfortunately, and a constant domain. So again, the C-terminal parts of these T-cell receptor chains are constant, the same between all T-cell receptors, but the N-terminal halves, well, maybe a little less than a half, have variable amino acid sequences, and they're highly variable, and it's the variation in amino acid sequence then that determines what antigen they will bind, and the antigen specificity. Again, the antigen binding pocket is made up of both the alpha and beta chains. So it takes both of these chains to form a single antigen binding pocket for a T-cell receptor. What immunologists estimate is that the body can make between 10 to 100 million different antibody molecules or T-cell receptors. Now, this is a startling conclusion, since I just got done telling you a couple of lectures ago that there are only about 20,000 different protein coding genes in the entire human genome. So how can the human genome be making 10 to 100 million different antigen receptor proteins, antibody molecules, or T-cell receptors? Well, the answer is in combinatorial joining. B cells and T cells, and only B cells and T cells, actually undergo DNA rearrangement. These are the only cells in the body that, as part of their regular differentiation program, rearrange portions of their DNA. So these antigen receptor genes are modular. They have a part of the gene that encodes the variable domain, and a part of the gene that encodes the constant domains. So they have a single gene that encodes the constant domain, okay, for each type of antibody or each type of T-cell receptor. But then the variable parts are broken up into 2 or 3 different segments. Okay? There's one segment called the V genes. There's another small segment, sometimes not all of them have these, but the heavy chain has D segments. And then they also have short DNA segments called J segments. Okay. To put a complete, and this is an example with the immunoglobulin heavy chain, to put a complete heavy chain together, the B cell selects at random one of these V encoding segments, one of these D encoding segments, and one of those J segments. And so the rearranged immunoglobulin gene then consists of a single V, D, and J selected at random from dozens of possibilities. And then this rearranged gene is the one that's transcribed and is eventually translated to make immunoglobulin, or the antibody protein. Then this is really the basis for how we can get antigen receptor diversity by combinatorial joining of different DNA segments. So here is a figure or a slide that explains how that works. So you recall that antibodies have heavy chains and light chains, so it takes a combination of one heavy chain and one light chain. Each heavy chain is made up of a combination of a V segment plus a D segment plus a J segment, and each light chain is made up of a combination of one V segment and a J segment. Okay. So let's say there are 100 different V segments and 10 different J segments, or 10 different D segments and 10 different J segments. If the cell can pick any V, any D, and any J, that gives us 100 times 10 times 10 is 10,000 different combinations. V, D, J combinations. So let's say there's 100 light chain V genes and 10 light chain J segments. That gives us 1,000 different light chain combos. Then remember, an antigen binding domain or an antigen binding surface consists of both the heavy and light chain. So we have an IgV or ID in immunoglobulin antigen binding surface consists of 10,000 heavy chains in combination with 1,000 light chains. So that's 10 to the 4th plus 10 to the 7th, or 10 to the 4th plus 10 to the 3rd. And that already gives us 10 million different possibilities. So you can see this combinatorial effects multiply very rapidly to very high numbers. And T-cell receptors undergo the same type of DNA rearrangements. Okay. The alpha chain has V segments and J segments that are combinatorially joined, and the beta chain has V and D and J segments that are combinatorially joined, and those have at least a similar range of different combinations. And then there are even more possible combinations, because when these are joined together, the joining is imprecise. When a V segment joins with a D segment, it can happen in actually any of in any reading frame, and then random nucleotides can be subtracted or added at the junctions. So because of all of these, we have an almost unlimited number of different possibilities in terms of antigen receptors. What happens then is that in each cell is undergoing each developing B and T cell in the process of differentiation undergoes DNA rearrangement independently and at random. So at birth, the body contains already a large population of T cells and B cells, and each cell has its own antigen receptor, okay, generated by this random combinatorial rearrangement with additional mutational processes that occur in this random joining. So you right now have in your body 10 to 100 million different T cells and B cells, each with its own antigen receptor. When a bacterium or virus invades your body, okay, and that's what we call an antigen, the binding of proteins or other molecules from viruses and bacteria to antigen receptors on certain of these B cells or T cells selects those cells to reproduce preferentially. Okay? And that's what we call clonal selection, because antigen binding selects an individual cell that contains a receptor that binds to the antigen, and then that cell with that antigen receptor begins to divide and produce a clone of itself. So mitotic division produces identical progeny. And so that's called clonal selection. And then these progeny can themselves be activated, and so you can get exponential increase in the numbers of that particular T cell or B cell clone. Oh, but wait. The virus or bacterium, these proteins can't directly activate T cells. T cells, the T cell receptors, are very particular. They like to have their antigens served up properly. And how is that? Well, the antigen has to be processed and presented. What that means is that these T cell receptors actually respond only to, or mostly only to, peptide antigens. So viral proteins or bacterial proteins first have to be digested, and they are degraded into small bits, small peptides, inside what we call an antigen presenting cell. And then these small bits are loaded onto MHC molecules. That's the major histocompatibility complex molecules. We'll just call them MHC molecules. MHC molecules are antigen presenting molecules. They bind these random peptides, they bind random peptide fragments, and they present them on the surface of the cell for inspection by T cells. So what are these MHC molecules? It's important for you to know that there are two different classes of MHC molecules. Class 1 MHC molecules are present in virtually all cells of your body, and my body as well. Okay? So they're present in virtually all cells. Okay? And they present internal antigens or internal peptides. So when your body's own cells turn over proteins, reach the end of their useful lives, they wear out, they get damaged, and then they go to the recycling center, they're called the proteasome. The proteasome chops them up into little peptides, and the peptides appear on the cell surface, loaded onto one of these MHC class 1 molecules. Okay? So My analogy is that a class 1 MHC molecule is kind of like the garbage can that every household has, and the household trash is put out in the garbage can. So MHC molecules essentially are displaying the cellular trash out on the cell surface. So every cell in the body does that. Class 2 are special MHC molecules present only in professional antigen-presenting cells. So APCs are antigen-presenting cells. And these are present on phagocytes, B cells, and dendritic cells. And they do pretty much the same thing, but they present external antigen. Well, how do they present external antigen? Well, because we're talking about cells that have, all of these cells have phagocytic activity, they internalize antigen from the outside of the cell into an endosome. The endosome fuses with a lysosome, and the lysosomal enzymes degrade these externally derived proteins into peptides, and then these peptides are loaded onto MHC class 2 molecules, and again displayed on the cell surface, on the surface of these antigen-presenting cells. Okay, so I hope you have straight the process of antigen presentation. These are peptides that are processed, meaning the proteins are chopped up into pieces or into peptides, and the peptides are loaded into MHC molecules and displayed on the cell surface. What's the point? Well, here we have a professional antigen-presenting cell displaying its endocytosed goods on MHC2 molecules on the cell surface. Along comes a helper T cell, or a CD4 plus T cell. So T cells come in two varieties, CD4 plus and CD8 plus. CD4 plus are helper T cells, and CD8 plus are killer T cells. So these are helper T cells, or pre-helper T cells, or naive, immature helper T cells, let's put it that way. And they remain immature until their T cell receptor binds an antigen or recognizes an antigen presented by an MHC class 2 molecule, at which point the helper T cell becomes activated, becomes a mature helper T cell, and it produces signaling molecules. They're called cytokines. So if you happen to run into that word, just know that cytokines are immune signaling molecules, immune system signaling molecules. So not only do they produce these signaling molecules, but they're stimulated to divide and produce lots of copies of themselves. Okay? And some of them become other helper T cells, and some of them become memory helper T cells. And these memory cells are long-lived. They can last decades in the body. So what's the role then of these helper T cells? So recall the antigen-presenting cells in association with a class 2 MHC molecule, okay, presents antigen, activates a CD4 plus T cell. Okay. And then we have B cells. Okay. Remember, B cells are the ones that can turn into antibody-producing plasma cells. And they have their own antigen receptors on their surface, which are these immunoglobulins. So let's say a B cell binds the same antigen from, let's say, the same virus. Okay? We have a virus infection. So the B cell internalizes that and presents it on its MHC2 molecule as well. Okay? So now we have an activated T cell, okay, and a B cell with its MHC2 presenting an antigen. The T cell and B cell talk to each other. The T cell recognizes the antigen being presented by the B cell, and it produces cytokines, the signaling molecules. Okay? And so these B cell and it's also going to bind antigen, the same antigen on its antigen receptor, is getting two signals. Its antigen receptor is binding antigen, and it's getting signaling molecules, the cytokines, from the helper T cell, and that then causes this B cell to divide, make lots of copies of itself. Many of them turn into plasma cells and produce large quantities of the same antibody that's on its cell surface, only it's a soluble form now. And then some of the progeny become memory B cells, which again have long lives and last decades in the body. And this is how the body mounts a humoral response, antibody production against antigens. What about cytotoxic T cells? The killer T cells. The killer T cells are differentiated from helper T cells because they have a different cell surface molecule called CD8. They also have T cell receptors. But killer T cells recognize antigen presented by MHC class I molecules. So remember, the class I MHC molecules are present in all cells of the body. Cytotoxic T cells are internal security or internal police. They're kind of like the FBI, and I guess, yeah, well, whatever. So they're internal security. Yeah. Their job is to go around inspecting the garbage cans of all the cells in the body, okay, and seeing what's in the garbage cans. So I remember I said class I MHC molecules are kind of like garbage cans displaying bits of cellular garbage, looking for anything suspicious. If the T cell receptor recognizes something that's foreign, okay, then the T cell becomes activated. Okay? And the activated cytotoxic T cell then produces molecules that do two things. Some of these molecules send death signals. Yeah. So they tell the cell to die. Okay? And just in case it doesn't die on its own, basically they turn on a process called apoptosis, which is cell suicide. Okay? So these death signals turn on cell suicide signals and try to induce the cell to commit suicide. If the cell refuses to commit suicide, it also produces other proteins that assemble in the target cell's membrane and create holes in it. Okay? And because of these holes, the target cell stuff leaks out, and the cell basically disintegrates. Okay? So that's our killer T cells. Now, how does this work? Or what's the advantage? The T cells, all the T cells circulating in the body, or most of the T cells circulating in the body, their T cell receptors don't recognize anything that is supposed to be in the body. So any self proteins, any T cells whose T cell receptors recognize self proteins or normal self proteins are eliminated during T cell differentiation. So the circulating T cells, their T cell receptors are primed to recognize only non-self or foreign proteins or foreign peptides. So any cell whose garbage is displaying something that can be recognized by a T cell receptor automatically means that it's doing something suspicious, something that is not supposed to be in the body. And that could mean that the cell has been infected by a virus, and so this is a bit of viral protein, or the cell has been subverted and become a cancerous cell. And since cancer requires mutations, then there may be mutant peptides that are displayed in the class I MHC molecules. And so it's the job then of these killer T cells to kill off any cell that is displaying anything highly suspicious. The last thing I want to talk about is go back to the role of helper T cells. Previously, we saw how the helper T cell helped to activate B cells. Well, the signaling molecules produced by helper T cells also help to activate the cytotoxic T cells, which means that helper T cells have a key role in assisting activation of both branches, the humoral branch and the cytotoxic branch of the immune system. And I want to close by saying that helper T cells, because of their CD4 protein, these are the targets of HIV infection. HIV selectively infects helper T cells and kills them off. And that is the source of the immune deficiency caused by HIV infection.